I'm Carter.
I'm Prachi.
I'm Jaden.
We're group four, and our keyword was anti-biofilm. So our article is about anti-biofilm activity of silver nanoparticles against different microorganisms.
The article that we chose is about scientists doing experiments on biofilms and using different concentrations of the silver nanoparticles to see what concentration level would work best in inhibiting the growth of the biofilm.
The type of nanoparticles used were silver nanoparticles.
In our article, the scientists conducted an experiment using silver nanoparticles, just like the ones that we had used in our lab. However, in this experiment, the silver nanoparticles were formed using a bit of a different method. First, with 0.169 grams of silver nitrate, or AgNO3, that was dissolved into 100 milliliters of deionized water. Next, gallic acid was dissolved into 10 milliliters of deionized water, and that was added into the silver nitrate solution under stirred conditions. The pH value was adjusted from 10 to 1.5 using nitric acid or HNO3, which caused the silver nanoparticles to flocculate. The resulting suspension was then filtered through a nitrocellulose filter with a pore diameter of 100 nanometers in a vacuum filter flask. The silver nanoparticles in the filter were then washed until a neutral pH was measured. Lastly, the silver nanoparticles were collected from the filter and sonicated to disperse them into deionized water and then diluted to the required concentrations for the experiment.
This is the application of the nanoparticles. To determine the antimicrobial activity of the silver nanoparticles, 100 milliliters of the nanoparticles were added to a polycarbonate membrane along with an agar medium and a culture of microorganisms. Another test run had different concentrations of the nanoparticles mixed into batches of microorganisms. In another test, the microorganism P. aeruginosa was chosen to determine the antimicrobial activity of silver nanoparticles against mature biofilms. Again, different concentrations of the nanoparticle were used in order to determine which amount would best kill the biofilm. Here's what the nanoparticles do.
So the purpose of the silver nanoparticles in this article was to kill the biofilms. Biofilms are caused by planktonic microorganisms that adhere to surfaces such as wounds, contact lenses, and mechanical heart valves, and initiate the growth of these microorganisms. Biofilms thrive where they have the proper water and nutrient levels, spaces to grow, and protection against environmental conditions. Biofilms are dangerous and are sources for disease outbreaks because they are on the surfaces of medical devices and can cause non-healing wounds and infections. This is one of the many reasons why silver nanoparticles are so important and useful. While they already are used in so many things, in this article they are examined to reduce microbial infections on the skin and even burn wounds. Experiments were also done to see how well and what concentration of silver nanoparticles are needed to stop the growth of biofilms before they even become a problem.
Here's some technological improvements for this application of silver nanoparticles.
Silver nanoparticles can and have been used to prevent the growth of harmful bacteria, diseases, and other microorganisms in biofilms. The biofilm protects these possibly harmful microorganisms from the harsh environments around them. Silver nanoparticles are able to destroy or otherwise prevent the growth of biofilms, giving microorganisms no way to protect themselves from the natural processes designed to kill them. Silver nanoparticles have great potential in the medical field, but not enough testing has been done for it to be applied practically and safely as of yet. They are currently used for food packaging, kitchen appliances, and pharmaceutical and medical nanoengineering.
This is our references page. The article that we used was Anti-Biofilm Activity, Silver Nanoparticles Against Different Microorganisms. And yeah, we're group 4, and our keyword was biofilms.